I'd like to introduce Dr. Jeannie Bailey, who will be moderating this session. Dr. Bailey is one of the core members of our research faculty. One of her areas of specialty is biomechanics studies around the spine as well as shoulder and knee. Thank you so much, Jeannie.
Hi, everyone. And we're going to let the speakers come up. This is paper session 2. It's going to be focused on basic science. And the first speaker is Dr. Yi, and her paper is called Effects of Glucocorticoids on Bone Mass Mechanical Properties or Molecular Perilacunar Remodeling Outcomes that are not rescued by PTH.
I would like to thank the organizing committee for providing me this opportunity to present my work. I have no conflicts to disclose. So glucocorticoids, we like using them to treating autoimmune disease and inflammation, and have been recently used for COVID-19 treatment. So imagine if you have a patient with severe COVID-19 who was treated with glucocorticoids. Well, that patient is now susceptible to GC-related skeletal complications, such as risk of developing secondary osteoporosis, 30 to 50% fracture risk, which is actually increased to 75% within the first 3 months of GC treatment, and is 9 to 40% more likely to have osteonecrosis or ON that can lead to the collapse of the joint, as illustrated in the scans of the femoral head. So here you can see that the femoral head has basically a very smooth surface, while the femoral head with ON actually have fragmented surfaces and osteonecrotic lesions. So with over 2.5 million people in the US taking GC for treatment, there is an increase in people being affected with these GC-related skeletal complications. And unfortunately, once a patient have GC-induced ON, it is irreversible and treatment options are limited. So due to inadequate options to prevent of reserved GC-induced skeletal disease, there is a treatment gap and a need for therapeutic intervention that prevent ON. So what if we can provide a prophylactic treatment that could prevent GC-induced ON to patients who are taking GC for their treatment? Well, this led us to wonder if osteocytes are a potential target intervention for GC-induced ON. So I'm going to give a little bit background about osteocytes. So osteocytes are embedded in the bones, and they create this network with their lacunae to communicate with other cells. And this osteocyte network is called the lacunar-cunicular network, or LCN. And the LCN regulate bone quality through local bone remodeling called perilacunar remodeling, or PLR. And PLR is a process where osteocytes express these enzymatic expressions, such as MMP13 and CAT-K, to resorb and replace local extracellular matrix to regulate this collagen organization and mineralization that contributes to bone quality. And this process is dynamic and adapts to both mechanical and metabolic changes. Our previous work have shown that GC-induced ON have suppressed osteocytic PLR that can lead to osteonecrosis. So here, GC directly acts on osteocytes and suppresses PLR activity with reduced PLR enzyme expression, disrupt LCN with the shortening of this caniculi, disorganized collagen, and matrix hypermineralization that leads to osteonecrosis. This suggests that osteocytic PLR plays a causal role in skeletal disease and bone fragility. So with the help from orthopedic surgeons, including Dr. Tad Vail, we were able to observe that patients with osteonecrosis have suppressed PLR that leads to joint disease and more cartilage degradation, revealing that osteocytes maintain joint homeostasis and that PLR suppression precedes and drives cartilage degradation. This led us to determine if we can rescue suppressed PLR with stimulating PLR activity back to homeostatic PLR, healthy PLR, and may lead to healthy cartilage and joint homeostasis. Therefore, we hypothesized that rescuing osteocyte-mediated PLR with PTH, or parathyroid hormone, will mitigate the progression of joint disease and GC-treated bones. So we determined whether combined GC and PTH will have beneficial effects on joint homeostasis and on osteocytes that can be used to guide clinical decisions. So to do this, we use female mice implanted either with placebo or prednisolone, referred to as GC, and injected with either vehicle or PTH. And some were treated with both. They were euthanized for further analysis examining bone mass, mechanics, and osteocytic PLR. So bone phenotype assessment using micro-CT on mid-cortical femur shows expected GC-induced bone loss and a modest elevated bone formation in PTH. Since suppressed PLR can affect bone quality independent of bone mass, we performed 3-point bending tests to examine the mechanical properties, and results showed similar patterns indicating GC bones are more fragile with this decrease in stiffness, and then PTH bones are less likely to fracture with this increased stiffness. The combined treatment group shows an overall bone loss, and mechanical outcomes also show an increase in bone fragility. And similar patterns were observed from other bone phenotype assessment and mechanical parameters, indicating that GC-induced bone loss and increased bone fragility is not mitigated by PTH. So to determine what is occurring molecularly, we examine gene expression using nanostring N-counter. This assay analyzes small amounts of RNA directly using fluorescent-coated custom-made barcode probes and is counted using the N-counter examining 96 skeletal-associated mouse genes. The volcano plot shows significantly upregulated or downregulated genes in gray. And RNA from osteocyte-enriched bones that are clean free from marrow unexpectedly show an upregulation in PLR-related genes indicated by GC, indicated in red. And this upregulation in PLR are observed in both the PTH and the combined treatment group, suggesting that GC-induced PLR gene expression is unaffected with PTH. So to elucidate if these effects are osteocyte cell intrinsic, we use osteocyte-like cells, MLOI4, treated with either GC, which was dexamethasone, PTH, or the combined treatment, and examine RNA expression. We observed decreased MMP13 and DMP1 expression with dex treatment, confirming that GC directly suppresses essential PLR enzyme MMP13 activity in osteocytes. We see a modest decrease of MMP13 and anticipated decrease in DMP1 and PTH is observed. However, in the combined group, PTH does not rescue downregulated MMP13 and DMP1 genes, revealing that PTH is unable to mitigate GC-induced suppression of MMP13 and altered bone remodeling related genes. So to determine if our outcomes of in vitro effects are the same as in vivo effects, let's go ahead and review. So we previously showed that short-term GC suppresses PLR, leads to osteonecrosis and joint disease. If GC-suppressed PLR can be rescued back to homeostatic PLR with PTH, this may lead to healthy cartilage and joint homeostasis. Therefore, can PTH mitigate the effects of GC on skeletal outcomes? In our study, GC results in an unexpected active PLR-related gene expression, bone loss, and bone fragility. PTH results in an anabolic effect with an active PLR-related gene expression, increased bone formation, and stronger mechanical properties. And the combined treatment results in active PLR, bone loss, and bone fragility. Therefore, PTH does not rescue GC effects, and differences in short-term versus long-term GC may have dual effects on regulating PLR. Further analysis is required to examine the joint. However, combined treatment fails to rebalance PLR and may exacerbate GC-induced skeletal diseases. So PTH may not be an effective strategy for mitigating glucocorticoids. However, other prophylactic treatments that target osteocytes may be better at preventing ON and joint disease. You'll be hearing about another potential therapeutic agent and its effects on regulating osteocytes by Dr. Gen Matsumai. And I would like to thank Dr. Allison, my lab members, and the funding sources that supported this study. Thank you.
Okay, the next talk is by Dr. Matsumai, and it's BGP15 can rescue bone quality by restoring osteocyte mitochondrial function.
Hello everyone, I am Gen Matsumai, orthopedic surgeon in Japan, and now I am postdoc at Ariston Lab of Orthopedic Surgery at UCSF. Today I would like to talk about my project titled BGP15 can rescue bone quality by restoring osteocyte mitochondrial function. Nothing to disclose. Bone fragility and fracture rates increase with age. Women in particular tend to have higher fracture rates than men due to changes in the hormonal balance after menopause. Bone strength is defined by bone density and bone quality. Although bone mineral density tests diagnose osteoporosis, it has been reported that half of patients with bone fragility have normal bone density. It is known that mechanical properties including bone toughness, post-yield displacement, and work to fracture decrease with age. Bone is metabolized by osteoclast and osteoblast, which defines bone density. On the other hand, osteocytes control bone quality through perilacunar canalicular remodeling, PLR, which Dr. E introduced in the previous talk. Therefore, to focus on osteocytes, to develop new therapies, we examine three features: bone quality, lacunocanalicular network, or LCN, and PLR gene expression. In the bones of young mice, the number of osteocytes is high and they are aligned. However, in the bones of aged mice, the number of osteocytes is reduced and their alignment is disorganized. The disorganization of the alignment is reflected in an increase in tortuosity. In other words, in addition to poor bone quality, LCN is impaired in aged bone. Furthermore, PLR-related genes, including MMP13 and MMP14, are known to be downregulated in the bones of aged mice. Since mechanisms by which aging suppress osteocyte function are unknown, we conducted RNA-seq on young and old mice, or old bone. We were surprised that our RNA-seq data reveals strong repression shown in the green of many mitochondria-related genes in aged bone. But is this repression due to a loss of osteocyte mitochondrial function? To answer the question, we collaborated with Dr. Glemsier to develop a powerful new two-photon microscope assay to quantify mitochondrial function in live bone. By visualizing bone embedded in osteocytes, we discovered that osteocyte mitochondrial function is significantly reduced in aged bone. Together, these data and our RNA-seq data implicate osteocyte mitochondrial function as an attractive target to improve bone quality. To target osteocyte mitochondrial function, we used the drug BGP15. BGP15 is currently in phase 2 trials for insulin resistance. Besides, BGP15 improves mitochondrial function in the setting of cellular stress and targets mitochondrial reactive oxygen species, or ROS, and other cellular functions. However, the effect of BGP15 on bone fragility has not been reported so far. We hypothesized that BGP15 can rescue bone quality through the recovering mitochondrial function in osteocytes. To investigate this study aimed to examine the effects of BGP15 on bone fragility using cultured cells and aged mice. First, we conducted in vitro experiments of BGP15 using cell culture. Excessive intracellular loss is associated with mitochondrial dysfunction and cellular senescence, and BGP15 has been reported to improve mitochondrial function and cellular function by suppressing excessive intracellular loss. Therefore, we tested the effects of BGP15 on osteocytes exposed to excessive loss. To test if osteocytes respond to BGP15, we used osteocyte cell lines, MLY4. To simulate cellular stress and loss production, we treated the cells with hydrogen peroxide. To test the cell intrinsic effect of BGP15, some cells were pretreated with BGP15 for 24 hours. After the hydrogen peroxide stimulation, we measured intracellular loss and performed qPCR for PLR genes. ROS was not elevated in the negative control, which had no stimulation, and in the group that received only pretreatment with BGP15, whereas ROS was significantly elevated in the group that received stimulation with hydrogen peroxide, as indicated by the black dots. Furthermore, in the group that received BGP15 pretreatment prior to hydrogen peroxide stimulation, the increase in loss was significantly suppressed, as indicated by the gray dots. Therefore, BGP15 can reduce cellular stress in osteocytes. We use the same model to determine if BGP15 can improve osteocyte PLR function. qPCR for PLR genes showed that BGP15 rescues osteocytes' intrinsic PLR gene responded to hydrogen peroxide and several stress with significant induction of MMP14. Since BGP15 protects osteocytes from cell stress in vitro, we decided to examine if BGP15 rescues age-related bone fragility using aged mice. In this experimental method using aged mice, 12-month-old female mice were orally administered 100 mg per kilogram of BGP15 for 2 weeks, followed by intraperitoneal administration of the same dose of BGP15 for 4 days. 2 days later, the mice were dissected and we evaluated bone mass, bone quality, LCN, and PLR genes. Micro-CT results in mouse femurs showing that BGP15 had no effect on bone mass. The results of a mechanical study on mouse femurs show that BGP15 significantly increased work-to-fracture and post-yield displacement compared to the control group. But does BGP15 improve aged bone quality by improving osteocytes? To answer the question, we performed the morphological evaluation of mouse femur cross-sections showing that BGP15 treatment significantly increased LCN density and decreased the tortuosity of osteocyte dendrites. Furthermore, gene expression analysis by qPCR was performed using RNA obtained from mouse humerus. The results show that MMP13, or gene related to PLR, was significantly upregulated by BGP15 treatment. To summarize the results of this study, BGP15 attenuates osteocyte stress and upregulates PLR-related genes. This could lead to an increase in LCN in bone, which results in an improvement in bone quality. I would like to acknowledge all of the researchers here. Thank you so much for your kind attention.
Next up, we have Dr. Davies on defining endogenous mitochondrial transfer in muscle following rotator cuff injury.
Thanks, Dr. Bailey. So I'm going to continue our theme of mitochondria here with this next talk, but I'm going to be focusing on a recent physiologic phenomenon that we found in skeletal muscle, specifically relating to cells being able to transfer their mitochondria to each other in the setting of injury. So our group has no relevant disclosures. So mitochondrial transfer is a phenomenon that we didn't really know existed 10 to 15 years ago. But what it refers to is basically cells directly transferring intact functional mitochondria to each other. And this is an interesting property that's been seen in a variety of different organ systems, including neural tissue, cardiac tissue, pulmonary tissue, and even bone, but interestingly hasn't really been looked at in skeletal muscle, which is one of the most mitochondrially rich tissues in the body. And so it raises lots of interesting questions about how we might be able to understand and then leverage this process to treating musculoskeletal disease. And so, when you have an interesting process like this, you want to find a good clinical system to look at it within. And it turns out that the rotator cuff seems to be an interesting clinical setting in which to study this process, because we already know from prior studies that there's mitochondrial dysfunction in the setting of chronic rotator cuff tears where you have a lot of fatty infiltration. And similarly, in controlled laboratory studies with acute tendon transection models, we've seen that there's an early amplification of the mitochondrial response in healing muscle. So it may be an interesting setting to look at this phenomenon of mitochondrial transfer. Now, within skeletal muscle, there are a variety of different cell types, including some mesenchymal stromal cells, which may be good candidates to try and assess this phenomenon in. And so one such group of cells are called fibroadipogenic progenitors, or FAPs. They're mesenchymal stromal cells that are distinguished by the surface marker PDGFR alpha. And based on their overall cellular profile, they're good candidates to potentially look at this process of mitochondrial transfer in. So we hypothesized that these cells may be able to donate their mitochondria to injured myogenic cells following a rotator cuff tear in mice. And based on other prior data suggesting that beta agonist treatment increases mitochondrial genes, we hypothesized that this process might be potentiated with beta agonist treatment. So to test this hypothesis, we bred mice that basically have FAPs that with mitochondria specifically labeled with EGFP, so they're labeled green. The genetics of these mice would be a talk in and of themselves, but basically the mitochondria within these cells are tagged with GFP, and then these mice underwent supraspinatus and infraspinatus tendon transection and suprascapular nerve denervation as a massive cuff tear model. We then performed a variety of downstream analyses. So we harvested the supraspinatus from these mice 2 weeks after injury. We used a variety of different microscopy techniques to look at the in vivo results of these tagged mitochondria from FAPs. We performed flow cytometry on these cells and then sequenced them using single-cell RNA sequencing to see what's going on in different subpopulations of these cells within the muscle. And then we performed co-culture experiments where we labeled FAPs with a green dye and then labeled C2C12 myoblast cells with a red dye and performed flow cytometry to quantify the amount of mitochondrial transfer that we see with and without beta agonist treatment. So just jumping into some of the pretty pictures here. So this is live confocal microscopy of our mitotag GFP labeled FAPs. So the FAPs are cells in green, and the green lines there are their mitochondria. And you can see them extending these cellular processes into adjacent C2C12 myoblasts. So this is what that process looks like at even higher magnification. In terms of our co-culture experiments, these FACS plots can be a little bit intimidating, but basically this is after we've sorted cells, after they've been co-cultured for 24 hours. We see that C2C12 cells labeled with our far red dye would show up in this quadrant. FAPs, which are labeled green, would show up in this quadrant. And then in this quadrant, we have C2C12 cells that have basically taken up FAP mitochondria. And so looking at our standard medium condition versus mirabegron, we see far more C2C12 cells that took up the labeled mitochondria from FAPs with mirabegron treatment. So all right, let's go forward. So looking at our single-cell RNA sequencing data, also very intimidating to look at, and you'll hear more about this technique from Dr. Garcia. But basically, these are all different subpopulations of FAPs that were sorted, and we can characterize them by their transcriptomic profile. And the interesting thing that we noticed is in this pre-adipogenic or sort of population of cells that seem to be differentiating down an adipogenic pathway, they expressed very high levels of mitochondrial structural genes, high levels of ATP synthase, and also high levels of the beta-3 adrenergic receptor. And moving into our in vivo data here, so again, these are the mitotag labeled mice. So this is a histological cross-section of the supraspinatus muscle. And we see the FAPs are going to be labeled in terms of their surface markers with the red, but the mitotag is actually the extent of their mitochondria. And again, we see almost an invasion of these cellular processes in green dye from the FAPs kind of going into the muscle fibers there. And when we quantified the amount of mitochondria in these fibers between the injured side of mice that underwent rotator cuff tear, the contralateral sham side, and the uninjured side, we saw something interesting. We saw a big increase in the amount of mitochondrial signal in both the injured and contralateral side, but very low intramyocellular signal in the uninjured side. And then, as expected, mice that did not have the gene activated with tamoxifen did not show a response. Looking at even higher magnification here, this is confocal and two-photon microscopy. This again just shows that both cells with nuclei in the interstitium, which we presume to be FAPs, as well as cells with nuclei kind of inside the myofibers, which are more likely to be myocytes, show an accumulation of these tagged mitochondria from FAPs. And then this is an even higher magnification, two-photon microscopy image of a FAP in the interstitium kind of extending these processes into the muscle. And when we looked at the overall level of ATP synthase expression between mice that were given the beta agonist mirabegron compared to mice that received DMSO, we saw a significantly higher ATP synthase level in the mice that received mirabegron. So just thinking a little bit about future directions here, we want to know if there's anything unique or special about these mitochondria from FAPs. So we go back to the single cell data, And we see, interestingly, there's one particular gene that stands out to us right now. So it's a gap junction associated protein, or connexin. And this is an interesting gene because in other organ systems, it's been associated with mitochondrial transport via interactions with microtubules, as well as potentially protection against ischemic type injuries to neurons. And so that may be kind of an interesting future direction. But to sum it up, these cells within muscle possess the ability to transfer their mitochondria to injured myofibers. This is the first study to look at this phenomenon in skeletal muscle, and then also to use that mitotag reporting system to follow the mitochondria in vivo. And future directions will look into this mechanism further and potentially evaluate the role of gap junctions in facilitating this process. So I'd like to thank all of my co-authors in the Abbott Society for sponsoring this event.
Okay, last on the list, Dr. Garcia on heterogeneous human fibroadipogenic cell subpopulations are altered in chronic injury.
Hi, everyone. Thanks to Michael for setting my talk up beautifully, but I'm going to talk a little bit more about these FAP cells and why they're interesting to us and how we looked at how they can do different things in human tissue. So I don't have any disclosures. So like Michael said, FAPs are cells that are stem cells within muscle, but they do good things, right? Like Michael said, they can activate satellite cells, which form new muscle. You get nice, healthy pink tissue if you take a pathology slide. But they can also do things that are not so good, right? In chronic injury, we get FAP cells that can turn into fibroblasts. They put down a lot of fibrosis. They can turn into adipocytes, put down a lot of fat. And this is not just in orthopedic injuries like chronic supraspinatus tears, but also in Duchenne muscular dystrophy and spinal issues as well. So I'm interested in how these cells decide whether or not they're going to be good or whether or not they're going to do these other deleterious things. And so the questions I have are, is this population just one cell that does all these things, or are these subpopulations of these cells that are heterogeneous and do these heterogeneous functions? And to do that, you can do a technique that Michael described called single-cell RNA sequencing. And that basically lets us look at the blueprint that these cells have. So we took muscle from human tissue, from supraspinatus, deltoid, and hamstring samples, and you can sort for FAP cells, and then through the beauty of this microfluidics chip, you get the transcriptome of all these single cells, which is basically about 20,000 genes from each cell and how much they're expressing each gene. And what we found is that these FAP cells are not just one cell type or one cell state. They exist in a number of different cell types or states, however you want to call them. And we think they perform these different functions. So there's a stem cell population, an adipogenic population, or a fibrogenic population based off the genes they produced. But that, like I said, that's just the blueprint these cells have. So how do we know that they're actually doing that? Well, we can look at the proteins they're producing as well. Another way to do that is to look at how many proteins they're expressing on their surface. So we can look at that with a technique called full spectrum flow cytometry. And what that lets us do is looks at all the combinations of those RNA molecules that we said, but now in protein form on the surface of the cells. And we looked at a number of different populations here, markers here, but what I can tell you is that we found individual cell states of these FAP cells that exist in these pre-fibrogenic, pre-adipogenic, or stem cell progenitor populations. And then we wanted to know, how do these populations relate to one another? So we can do a computational modeling technique where we find a pathway from each cell type to another. And we see that we found a pathway from a progenitor cell that can lead to an adipogenic cell, or to a fibrogenic cell, or even possibly a tenogenic cell. So to tease that out, we put FAPs in culture, and we put them through different media conditions, sort of like how Michael described as well. So you can put them in a standard growth media, or adipogenic media, or fibrogenic media for 5 days. And then we can do staining with those surface markers that we know mark these different populations. And we see that in the adipogenic population, CD10, which marks this adipogenic population, is bright green, so it's much upregulated, where you don't see that in the fibrogenic population and in the fibrogenic media. And then conversely, for CD55, which we thought would mark the fibrogenic population, we see it only in the fibrogenic condition. So that tells us that we're onto the right track here. And that's just showing that from the progenitors, we get to these two different fate decisions. So now we can look at, so we know that in chronic injuries like supraspinatus tendon tears that you can get fatty degeneration like you see here in this Gutelier sort of stage 2 muscle, and where we can compare it to healthy muscle where they don't have this injury. And what we found is that the similar volcano plot that you saw before is that these cells have more progenitor markers like Thy1 and activation markers like FOS, which are genes that the cells are doing things like being activated and not quiescent stem cells. And then also interestingly, there was a big decrease in this secreted protein called DLK1, and we were interested in following that up. So what we looked at is that we took more human muscle from injured and healthy tissue and did this full spectrum flow cytometry. We can look at all the different markers again. And we found that actually DLK1 protein is decreased as well. So in healthy tissue, there's 15% of the cell FAPs that express this marker, but in full tear patients which have fatty degeneration, there's only about 3%, and that was significantly different. And we know from prior literature that DLK1 can suppress fat in adipose tissue, but it's never been looked at in muscle. So we took FAPs and added either just controlled PBS in culture or with recombinant DLK1 human protein and put them into adipogenic stimuli media, and we see that in the DLK1 treatment that there's much less fat formation, and we think that's because there's much less, or not much less, but a little bit less adipogenic progenitors as well. So what this is getting at is this kind of an updated model of how FAPs exist in homeostasis and in injury. The population is heterogeneous. It's linked to function, we think, and that function is altered in injury, and we see an alteration and loss of DLK1, which may allow cells to proliferate and to form more fat. And of course, this isn't done by just me, but so I want to point out Augustine, Hannah, Justin, Miguel, who I don't have a picture, Michael, Shue, and Brian Feely, who have all helped me greatly with this work. Thank you.
We have time for a question or two, and I'll start. Dr. Davies, I have a question. It might be naive. When you're looking at these things following injury to the rotator cuff, how exactly are you inferring directionality of whether or not it's degenerative or recovery? Because you're just looking at how the state of these cells in a certain time point following injury. So what are you doing to delineate that?
Yeah, absolutely. It's a good question. So I think, first when you're generating a new animal injury model, you have to kind of follow it out over time and see what eventually happens. And so we know with our mouse rotator cuff injury model, we start to see a lot of fatty infiltration and fibrosis around 6 weeks or so, and then it just continues to progress kind of from there. Truthfully, that's within sort of the time point that we're normally using these animals for experiments. So we haven't necessarily followed them out for like 6 months after tear to kind of look that long, but we see that they develop the pathology that we see in humans at about 6 weeks. And so if we're looking at an earlier time point, like 2 weeks, going back to your question, I think you're asking whether the transfer that we're seeing is part of the degenerative process versus regenerative. Mm-hmm. So, to answer that, we've looked in other models where you can kind of clearly see hallmarks of myofiber regeneration, so centrally nucleated myofibers, and we see that the mitochondria seem to preferentially localize within those fibers in a different injury model. But I think from that, we're sort of inferring that it is part of the regenerative process that we're seeing this.
Any other questions? If not, I think we have to move on to the next talk, which is Dr. Feely, wherever he is.